Hi class, welcome to the next lecture of the Medical Parasitology Lab. Today we're going to be talking about gut, lung, and blood trematodes. So trematodes are a parasitic class within phylum Platyhelminthes. Platyhelminthes is known collectively as the flatworms, and trematodes themselves are referred to as flukes. So there's other classes of parasitic Platyhelminthes, such as cestodes and monotogena, but today we're just going to be focusing on trematodes. So trematodes are unsegmented and they're acoelamates, meaning that they have no body cavity and they have no specialized circulatory or respiratory system. So instead, oxygen and nutrients just diffuse through their skin. And the vast majority of trematodes just have a digestive cavity that only has one opening for both ingestion and both for egestion, or the removal of waste. So they have only one opening for eating and for excreting waste. So moving on to the general anatomy of a trematode. So all of them have an oral sucker, which is what is used for their mouth. They also have what's called a ventral sucker, or also known as an acetabulum, and that's what they use to attach to the host. And all trematodes are known as hermaphrodites, meaning that they have both male reproductive organs and female reproductive organs, so they can fertilize themselves. And in this particular example, this trematodes has an excretory pore that it can use to remove waste, but most of them don't have this. So here in this image we can just see the general anatomical structures that you'll see within a trematode, such as the female reproductive system, such as the uterus, the yolk glands, which produce yolk cells for the egg, and the male reproductive organs, such as the testes. In this example, they're branched. So moving on to the general life cycle stages of trematodes, we'll talk about more of these in more detail later. So the ones that say always present, it means that they exist within all species of trematodes, where some of them don't exist in all species of trematodes. So we have the miracidium, which tends to exist in the external environment and can swim around in water using their cilia. We have the sporocyst and the retia, which tend to exist within the intermediate host, such as a snail. We have the cercariae, which penetrate the skin of the definitive host. And then we have the metacercaria, which is a cyst that contains a juvenile, which is found within the skin of various hosts, such as fish. So moving on to our first parasite of today, it's Fasciolopsis buski. So this is the largest intestinal fluke of humans, and it's the causative agent of fasciolopsiasis. So the eggs are about 130 to 150 microns in length, while the adult worm is about 20 to 75 millimeters in length, and its intermediate host is the snail. So looking at the life cycle of F. buski, so the metacercariae can be in contaminated water or contaminated plants, and those will be ingested by a human or a pig, and that's what will cause the infection. So the metacercariae exist within the duodenum, and then the adult worm then colonizes within the small intestine. So then the adults lay on embryonated eggs. Those are passed within the feces. It takes about 7 weeks for the eggs to become embryonated in warm water. Once they're embryonated, they can then hatch. They produce miracidia. The miracidia can swim around in the water, and they penetrate the skin of a snail. And when they're inside of the snail, they undergo further development from a miracidia. They then become a sporocyst, to a retiae, to a cercariae. Afterwards, the cercariae then are excreted from the snail. The cercariae can freely swim around within water, and eventually the cercariae encyst within the water, and then they become this metacercariae form, and then the cycle repeats itself. So here we have the anatomy of F. buski, the adult worm. So, as you can see from their hermaphroditic nature, you have branched testes, you have the ovary, you have the uterus here in dark. It's dark because it's full of eggs. You have the ventral sucker, or the acetabulum, that they use to attach to the host. And then there they have the oral sucker, which contains the mouth. All of this here, the gut, is their intestines. They have this Melas gland, which is what makes the materials to make the eggshell. And then the vital area is what produces the yolk cells to make the yolk of the egg. So here is the egg of F. buski. It takes about 3 to 7 weeks for it to become embryonated in warm water. In a non-embryonated egg, you'll see the yolk cells inside the egg, and then over time those will dissolve and start to become the meracidium. And up here is the operculum. That's what opens up and allows the meracidium to escape when it's hatching. It actually kind of flips open like a bottle cap. And so here's an image of the meracidium. They have cilia that they use to swim around in the water, and they penetrate the skin of a snail host in order to enter the snail. There's a video of an F. buski egg hatching. So moving on to the next parasite of today that looks pretty similar to F. buski is Fasciola hepatica. So this is the causative agents of fasciolisis in humans. It's also known as the common liver fluke or the sheep liver fluke, and the adult is up to 30 millimeters long by 15 millimeters wide. So here's a life cycle of Fasciola hepatica. The Metasecariae are contained on top of vegetation within the environment. So a host then has to eat the contaminated vegetation in order to become infected with the parasite. So the Metasecariae exist within the duodenum, and then they migrate through the liver to the biliary ducts, and then so the adult parasite actually colonize the liver within the bile ducts. So the way they enter the liver is they're actually able to break through the intestinal wall and then travel to the liver. So the eggs will be released within the feces. They're also unembryonated when they're released from the feces, and it also takes them a few weeks to become embryonated, where they will then hatch and produce the meracidia. The meracidia swim around in water, and then they penetrate the skin of a snail. Inside of the snail, they undergo development until they eventually become the cercariae. The cercariae then escapes from the snail, and when they're in the water, they then insist to form the Metasecariae, and then this infection just repeats itself. And another mammalian host that they can infect is cattle and sheep, so that's also why they're known as the sheep fluke. So here's an egg of Fasciola hepatica. So the eggs are very difficult to distinguish from Fasciolopsis buski, so I won't ask you on the test to differentiate between the two eggs because they're pretty difficult to tell apart. So they both have an operculum. Something that can be used to differentiate Fasciola hepatica from F. buski would be the abopicular end, which is the end that is opposite of the operculum. It tends to be roughened or irregular in shape, but it's not really visible through a microscope of this magnification. You would usually need a higher magnification in order to tell. So here is the meracidium of Fasciola hepatica. It also looks pretty similar to the meracidium of F. buski. So here's a comparison between the Fasciola hepatica and the F. buski adults. They look pretty similar, except that F. hepatica has a more pointed anterior end than F. buski does. So that's the best way really to tell apart the adult forms. Moving on to our next parasite, known as heterophyes. So the heterophyes are small trematodes. They're usually under 2 millimeters long, and their outer surface is protective in that it's covered by tiny spikes. So this worm lays embryonated eggs, and they contain the meracidium already fully developed, so they don't need time to become embryonated in the water. They're already embryonated when they exit the host, so that means that the eggs are infective immediately. So here's the life cycle of heterophyes. So a host will become infected by ingesting undercooked fish containing the Metasecariae. the Metacercariae will be within the skin of the fish. So what happens in a human is the Metacercariae then exist within the small intestine, and they eventually grow into the adult form within the small intestine. So eventually the adult will produce the embryonated eggs, and those exit the host through the feces. And again, they already contain the miracidium already fully developed. So these eggs are immediately infective when they exit the host. And so a snail will eventually ingest these eggs, and the metaceria emerge from the eggs, and then they penetrate the snail's intestine. They then develop into cercariae, and those are released from the snail. The cercariae will then penetrate the skin of a fish, and then they will eventually encyst back into the Metacercariae form within the skin of the fish, and then the cycle just repeats itself when undercooked fish is then ingested by a human or another mammal. So here's a more detailed image of the heterophyes adult. So here we have the oral sucker, or the mouth area, the pharynx, the intestines, the acetobulum, or the ventral sucker that it uses for attachment, and here we see eggs within the uterus. This image also shows the testes better, number 6. Number 4 would be the vitellaria, which produce the yolk cells for the eggs. Number 5 here would be the ovary. And you can find these labels in the notes of the PowerPoint, so you can refer to them later. So moving on to the next parasite of today, Metagonomus yokogawae. So it has the same life cycle as heterophyes in that it involves snails and then fish, and then the human has to then eat undercooked fish in order to be infected. And they also resemble heterophyes in terms of their morphology. However, something that is really a good way to distinguish the two is the position of the ventral sucker, or the acetobulum. In the instance of this parasite, it's referred to as a genitoacetobulum because it's a little bit lower down than it is in other trematodes. So in the case of Metagonomus, it's a little bit off-centered and more closer to the genital pore, where in the previous parasite, it's more centered. So you'll see here, the acetobulum is very off-centered in this parasite, so that's what helps differentiate it from heterophyes. Here it still has all of the other same structures, like the oral sucker and the pharynx, the intestines here, eggs within the uterus, the ovary, and the testes. So moving on to the next parasite, Clinorchis sinensis. You might see it as Opisthochus sinensis. That's what it used to be referred to, but they discovered that it has more branched testes than other parasites, so they gave it its own genus, which is Clinorchis. So this is known as a Chinese or Oriental liver fluke because it has a high prevalence in Asia, and it is the causative agent of the disease Clinorchiasis. So here is the life cycle of C. sinensis. So similarly to the other parasites that we talked about, the Metacercariae will be within the flesh of freshwater fish. So then a human then has to eat the undercooked fish meat in order to become infected. So the Metacercariae will exist within the duodenum, and the adults will infect the biliary duct of the liver. And so this parasite passes embryonated eggs, meaning that they're immediately infective. So then the eggs then have to be ingested by a snail, and the snail acts as an intermediate host and will become infected with the parasite. So the eggs then produce miracidia, and then those mature eventually into cercariae. The cercariae then exit the snail. They swim freely in the water, and then they eventually will burrow through the skin of a fish, and the cycle just repeats itself. So here is an egg of C. sinensis. So something that makes this egg unique is that it has what's referred to as shoulders surrounding the operculum. So that's something that is used to identify this egg. It also has what's called an abopicular knob at the abopicular end. And these are overall just very defining features of this egg that can help you to identify this egg. So here's the adult of C. sinensis. So like I mentioned, it has the branched testes. There's the vitellaria. The uterus is here. the ceca, or part of the intestine, the oral sucker, the pharynx, and then the ventral sucker, or the acetabulum here. So here's a good image of the metacercaria, which you'll find within the skin of the fish, and they contain the immature form of the fluke. So here's just a detailed diagram showing what they look like. These will eventually exist and release the mature adult form of the fluke. So moving on to the next parasite, it is Paragonimus westermanni. So this is the most common lung fluke. It's the causative agent of paragonomyosis. So both eggs and adults can be found within the lungs, and its symptoms can be confused with tuberculosis. If you're familiar with tuberculosis, it causes granulomas within the lungs, which is just a bunch of scarred tissue and immune cells. They form this large cluster when they're trying to attack the parasite, and they form these granulomas in the lung, and that's something that also happens with tuberculosis. So overall, they can have similar symptoms with a lot of coughing and these granulomas. So here's the life cycle of Paragonimus westermanni. The cercariae will invade a crustacean. They insist into the metacercariae. So the metacercariae will be within the crustacean, such as shrimp or lobster. So the human then has to ingest undercooked crustaceans. So the metacercariae exists within the duodenum. So the adults can stay within the intestines, or they can actually burrow out and escape the intestines, and they tend to travel to the lungs. They can also travel to other organs, but the lungs is usually what they prefer. And so when they're in the lungs, the way that this parasite is diagnosed is you can actually find the eggs within the sputum, or that's like the phlegm that you cough up when you're sick, so you'll actually cough up the eggs. Or if the parasite remains within the intestines, the eggs will also be passed in the stool. So they pass unembryonated eggs that take a few weeks in warm water to become embryonated. So the Myristidia hatch from this egg and they penetrate a snail. They mature within a snail to produce a cercariae form, and then the cercariae then invade the crustacean, and then the cycle just repeats itself. So here's the adult form of Paragonimus westermanni. So we can see here the intestines, the ovary, the testes, and the vitellaria. And then here they do have an excretion duct, so they do have a separate hole for eating and for excreting waste. So that's pretty rare among trematodes, so this is one of the few trematodes that actually has an excretion duct. So here's an egg of Paragonimus westermanni. Here's the operculum, the abopercular end. So they tend to be brown in color. On the sides, the wall tends to be thin, and then at the abopercular end, it gets thicker. So that's something that can be used to identify Paragonimus westermanni's eggs. So moving on to our next parasite of today, and probably one of the more interesting looking ones of the trematodes, is Schistosoma. So Schistosoma are blood trematodes, and they're responsible for the transmission of schistosomiasis. So there's three main species that infect humans. There's Schistosoma mansoni, Schistosoma japonicum, and Schistosoma hematobium. And something that's unique about hematobium is it's the only Schistosoma parasite in which the eggs will be found in the urine. The other Schistosoma species will have the eggs only found within the feces. Okay, so here is the life cycle of Schistosoma. So the eggs are found in water. They're either shed through the feces or through the urine in the case of Hematobittum. So the eggs hatch in water and they release the Myristidia. The Myristidia then penetrate the skin of a snail. The sporocysts and the cercariae then develop within the snail. The cercariae then released into the water. The cercariae can then penetrate through the skin of the human. That's how they infect the human. Once the cercariae are inside of the skin, they lose their tail and they become schistosomulae. So the schistosomulae then circulate throughout the blood and throughout the body. They eventually reach the portal blood of the liver, and that's where they mature into the adults. So the adult worms will then migrate to the mesenteric venules of the bowel or the rectum, and that's where they lay their eggs that circulate to the liver and then eventually are shed in stools. Or they can go to the venous plexus of the bladder, and then their eggs will be shed in the urine, which is the case of Hematobittum. And so if this cycle just repeats itself, and the infection will be perpetuated. So here's a comparison of the schistosoma eggs. You have Hematobittum has a sharp terminal spine at the end of the egg. S. mansoni has a very large, very sharp lateral spine that's kind of pointing out at the side of the egg. And then Japonicum has a very small, rounded lateral spine that's very tiny and sticking out here. And the exact function of the spine isn't known, but it's thought that the eggs can cluster together using the help of the spine. So here's just a more clear comparison of the spines. There's S. mansoni with the large lateral spine, Hematobittum with a terminal spine, and then sometimes Japonicum doesn't even have a spine. So that's just a very clear comparison between them all. And so here's a Schistosoma miracidium. This is the stage that hatches from the egg and swims around in the water and will eventually penetrate the snail. So they swim around using cilia, and this structure at the anterior end is what helps them penetrate the skin of the snail. And here's a few videos showing a miracidium hatching, and then showing how they swim around. So here we have the sporocyst that's found within the snail. The germinal cells here will give rise to the sporocysts, and they undergo at least 2 generations of multiplication until they give rise to the cercariae. And here we can just see several important organelles and structures of the parasite labeled. So moving on to the cercariae, this is the form that will penetrate the skin of the human. So they do have an acetabulum, similarly to the other parasites that we talked about today, to help them with attachment. This duct opening in their heads is what they use to help them burrow through. And they just have other organelles to help them survive within the environment of the human. And this is a pretty interesting picture. This was actually a histological section of the skin of a mouse, while the cercariae was actually burrowing through the skin. So it's entering the skin through a little hair follicle. So here you can see the hair. And then so there's the tail as it's swimming in. There's the head of the cercariae. And then here, all of these different layers. Those are just the different layers of the skin. And then the sebaceous gland is just an oil-producing gland. It's usually located near the base of a hair. So overall, that's just a really interesting and detailed image that kind of amazing that they were able to take that image as the Cercaria was actually burrowing inside of the skin. So that shows you, I guess, how small they are, that they're able to purposefully take advantage of the little hair follicle, that little tiny hole where the hair grows from, and that's where they burrow through. So then here's a more detailed image of the schistosomule. This is just what the parasite is called once the Cercaria loses its tail, and they always lose their tail when they penetrate the human skin. Here you can just see a light microscope image of them, and then a more detailed electron microscope image of them. And so here's the adult worms. Here on the left we have the male, and on the right we have the female. Here is the intestines spanning through the entire worm. The female also has the intestines spanning through the entire worm. The female also has the vitellaria, which it uses to produce the yolk cells for the egg. And so here's what the worms look like when they're in copula. That means when they're paired together. So when they're in copula or paired together, they're considered hermaphroditic. So remember how all trematodes are considered hermaphroditic? So this is why they're also considered trematodes and hermaphroditic. And when the parasite and when the adult parasites are circulating through the blood, you're never going to find a male or a female alone. They're always going to be in copula together. So the female is the thinner and the darker one. She resides within the gynecophoral groove of the male. And then the males are larger, but they're also covered in these little tubercles or bumps on their surface, possibly for protection. And here's just an even more detailed microscope image of the incopula worms. So the reason why the female is a lot darker is this is due to blood cell digestion byproducts. Hemozoin is made from hemoglobin degradation. So here you can see the female has similar organelles or structures to what we saw earlier, such as the Meles gland, the esophagus, and the intestine, just going down the entire length of the parasite, the vitelline glands or the vitellaria, and then her uterus will appear dark when it has eggs inside of it. And in the male we have the gynecophoral canal, which is what the female uses to attach herself into. Alright, so here is a table summarizing all of the different eggs that we covered today and showing just a good comparison between them. So here we have Schistosoma japonicum. Sometimes it has a little knob spine, sometimes it doesn't. Schistosoma mansoni always has that large lateral spine. Schistosoma hematobitum always has the terminal sharp spine. Paragonimus westermanni is that one that looks brown, and it's thin on the sides, and then as you get closer to the abopicular end, it gets thicker. And then Fasciola hepatica and Fasciolopsis buski are really hard to tell apart just through microscope images, so I probably won't ask you to differentiate those two. But differentiating these from any of these other ones is fair game. And then we have the smaller eggs from Clinorchis, from Heterophyes and Metagonomus. And Clinorchis is that unique one that has an abopicular knob. You can kind of see it here in this image. And then it also has shoulders. And with that, that's the end of today's lecture. I hope you learned something new, and I hope you have a good day.